Hi everyone, this is Kiranjeet from section 706. Khushpreet and I performed an experiment using Beta vulgaris or beetroot in common terms, testing its preservative properties against foodborne spoilage microorganisms Escherichia coli and our agenda for this presentation is to introduce why we used Escherichia coli and Staphylococcus for this experiment, why we used minimum inhibitory concentration determination test, and as we know, people are so interested in eating meat in North America. That's why it was of extreme interest to test the preservation of the meat products. For this experiment, we chose beetroot to test for its preservative properties. The reason for choosing beetroot. The hypothesis of this experiment was to show that the ethanolic extract of Beta vulgaris is effective against common human infections in terms of antibacterial activity. It is predicted that Beta vulgaris extract has dose-dependent antibacterial activity against both gram-negative and gram-positive bacteria, and that it can be used in place of sodium and potassium nitrites to preserve meat and their products.
Hello everyone, my name is Khushpreet Kaur and today I will explain. So as you guys know, in this experiment we are trying to find out the antimicrobial activity of beetroot, and for this we need beetroot extract, and for extraction we used a method called ethanol extraction method. For this method we firstly took 4 medium-sized beetroot and then cut them into a 4 pieces or fine pieces and then put these. So for extraction, after shaking our extract for 24 hours continuously, the extract was then passed from filtering funnel to filtrate, and so in order to explain the antimicrobial activity of beetroot, the first method that we had used was minimum inhibitory concentration, also called MIC. So for MIC, we first prepared our media from MHB and we totally prepared 12 tubes of media, in which the first tube was double standard because the amount of MHB was double in it as compared to the rest of the 11 tubes, and these tubes were called single standard. And after preparation of our media in all of the so in order to perform minimum bacterial concentration, we use the tubes from MIC. So after performing MIC, we got the MIC value for E. coli 1 by 64 and for S. aureus 1 by 256. So these were the tubes in which there was minimum inhibitory concentration, and we used these tubes for our MBC. So in order to perform the MBC, So proceeding further, our next test was agar-well diffusion, in which firstly we took two MHA plates and then swabbed our both of the organism E. coli and S. aureus on each plate. And after swabbing, we make four wells in our agar because we used. So the last test that we had performed was epicillometer test, also called E-strip test, which is just a replica of finding the antimicrobial activity of beetroot extract. So first of all, E-strip was prepared for this, and for the E-strip preparation we use filter paper, Whatman filter paper 1, and then firstly the circular filter paper was cut into a. So after pouring the 6 different concentration of beetroot extract in 6 different column of E-strip with the help of a micropipette, we then took 2 MHA plate and then swabbed the first plate with E. coli and the second plate. So talking about the results of MIC and MBC, in case of MIC, E. coli had minimum inhibitory concentration at 1 by 64, which means that the beetroot extract stopped inhibitory activity after this dilution. However, for S. aureus, the.
For agar-well diffusion method, MHA plates are used as MHA is a medium that supports the growth for non-fastidious bacteria and is also low in sulfonamides. The holes in 4 mm thick agars were made using pasteur pipette to pour different concentrations of beetroot in the wells. The plates were swabbed with the cultures of E. coli and S. aureus that were inoculated in nutrient broth 24 hours ago before swabbing on the agar plates. After swabbing, the wells were filled with beetroot extract and the inverted plates were kept for incubation at 37 degrees Celsius for 24 to 48 hours. The expected results after 24 hours was to have a zone of inhibition around the wells according to the different concentrations of beetroot. However, for this experiment, the expected results were not obtained by us. There was no clear zone of inhibition around the wells as it can be seen in the picture that is attached to the slide. The reasons for not getting the zone of inhibition as expected can be many, which are explained in the discussion slide ahead. Another unique method used by us for testing the antimicrobial properties of beetroot was E-strip or epsilon strip test. This strip is similar to Kirby-Bauer disc diffusion method. The only difference is that for E-strip, a strip of different concentrations is placed on MHA, while in Kirby-Bauer technique, different discs of specific concentrations are placed on the same plate. This discussion slide covers the reasons for not getting desired or expected results for agar-well diffusion method and for E-strip test. The two possible reasons for agar-well diffusion method can be as follows. Number one, the concentrations of the cultures of E. coli and Ascaris that were used to swab on MHA were not standardized to 0.5 McFarland standard. The other reason can be that the agar was too thick that it resulted in false resistant results around the wells. For the E strip, we could have used pour dry pour method, which means that after pouring the beetroot concentration. Let's talk about the summary. This presentation was all about the beetroot preservative test, in which we used MIC, a Garwell method, and a very unique method of testing antimicrobial properties of beetroot, that was epsilometer test. Unfortunately, two of our tests failed, the reasons for which are discussed above. We are waiting for your valuable feedback, and thank you for watching till now.